Good morning, good noon, good afternoon to everybody. I'm happy to announce the next European Society of Mycobacteriology webinar. And today we have a special guest, Jakko van Ingen, which most of you do know, but I do give a quick introduction for those who are less familiar with him. So Jakko van Ingen is a consultant clinical microbiologist and head of the Mycobacteriology Reference Laboratory at RaboudUMC Medical Center in Nijmegen, the Netherlands. He has authored over 200 papers on mycobacterial disease in peer-reviewed scientific journals and is consulted on diagnosis and treatment of tuberculosis and non-tuberculous mycobacterial disease from all over the world. His research group focuses on the pharmacodynamics of antimicrobial drugs and the development of new treatment regimens, both for TB and non-tuberculous mycobacterial disease. So he's well renowned on the non-tuberculous mycobacterial diseases. So welcome, Jakko. Regarding some housekeeping notes for the current webinar, you can send us your questions during the talk using the live chat window. So to use the live chat, you will need to sign in to YouTube with either a YouTube or a Google account and then join the channel. So live chat messages are not private, everyone can read them. So be careful what you write. And please include your affiliation in your questions so we can get back to you and address you when we pose the questions. The chair will ask the speaker audience questions after the talk. So the sooner you ask your questions, the more likely the speaker will have time to answer it, because Jakko indeed has to leave strictly at 3 o'clock to be present for a next webinar. So if you have not been able to ask your questions via the live chat for any technical reason, you can also email them to the office email address that is written down here. And the webinar will also be recorded and will be available later on via YouTube. And last but not least, these webinars are brought to you in collaboration with our sponsor, Becton Dickinson, for which we are grateful. They, however, do not have any say in the content of the presentation. So Jakko, the floor is yours.
Well, thank you very, very much, Lane, for that very kind introduction. It's always a little humbling, you know, when you're introduced by someone who is a giant in mycobacteriology. So, yeah, my name is Jakko van Ingen. I work at the Radboud University Medical Center in Nijmegen, the Netherlands, and I'm extremely grateful to be invited by the European Society for Mycobacteriology. And I have to say, I do miss the live conferences because ESM conferences are the best there are, both in terms of content and in terms of the people attending them. But well, instead, we're doing a series of webinars. So today's webinar is a sequel. So the first part of the series, I also did one on NTM that was focused on drug susceptibility testing and treatment, so antibiotic treatment of NTM disease. So today I will not be touching upon those subjects much. The focus of today will be slightly different, and it is moving towards a deeper understanding of what is NTM disease and how should we think about diagnosing and treating it, and what's newly emerging in this field that will change our current dogmas in the upcoming few years. So I have to show, of course, my financial disclosures, which are not that relevant as we will not be discussing antibiotic treatment, which is where most of the consultancy work that I do is focused on. So at Radboud University Medical Center in Nijmegen, the Netherlands, we have a dedicated program for treatment of patients with NTM infections and tuberculosis. So we are a reference clinic focusing on difficult to treat TB cases, including drug-resistant forms of TB, as well as patients with non-tuberculous mycobacterial disease that we see within a multidisciplinary team consisting, of course, of pulmonologists and infectious disease physicians, but also dedicated pharmacists with in-depth knowledge on anti-TB drugs, dedicated radiologists, as well as clinical microbiologists like myself, focusing on diagnostic mycobacteriology. And we are also a trial site for clinical trials, and we have a research lab where my group works on finding new treatment regimens for TB as well as for NTM disease. So where are we at? It's 2021, and we've by now learned that NTM-PD, or pulmonary disease caused by non-tuberculous Mycobacteria, really comes in two important manifestations: the fibrocavitary manifestation, which is sort of similar to pulmonary tuberculosis, although it develops over a longer time course and it affects a slightly different patient population. And the second, and maybe more frequent in some parts, manifestation, the nodular bronchiectatic disease, which again affects a very different patient population, is also radiologically very distinct, as you can see in the CT scan at the bottom, where there's a combination, as the name suggests, of nodular lesions or bronchiolitis and bronchiectasis. So in most studies, the prevalence of these infections seems to be on the rise. And that is very worrying, because at this point in time, we do not have very effective treatment regimens to treat these infections. Cure rate at this point in time for the most common species, like those of the Mycobacterium avium complex, are in the 50 to 70% range. And for Mycobacterium abscessus, which is also quite frequent, but an absolute nightmare to treat, this is as low as 40 to 50%. And even those that you've been able to treat successfully stand a very high chance to get or to suffer from a recurrence of the disease. And like in tuberculosis, recurrence can mean either the original infection not being eradicated and coming back, or to get reinfected by a new strain, perhaps of the same species or a different NTM species, and then get sick again. And later on in the talk, we'll get back to that topic and show that, in our current era, with the introduction of sequencing techniques and the spread of those techniques, I think we should work on making that distinction between reinfection or relapse a standard practice in diagnostic mycobacteriology. So we'll get to that later on. So when discussing the part 2 of the NTM seminars, we thought about several topics and we decided that today we should focus on a question that we get posed a lot. Is it possible to distinguish TB and NTM disease clinically? We'll touch upon that first. Then, in conjunction with that, I will focus on the microbiological diagnosis of NTM and discuss, so what's new in the lab and how does that affect clinic for identification of NTM as well as for susceptibility testing. Can we move towards whole genome sequencing like has happened in TB? Or are we just not there yet for NTM? Or maybe is it a different universe altogether and are the rules so different that we might not be able to ever apply it? And that will feed into the third and last part of today's session, where I will be discussing evolution and mutation in NTM. So how can we use molecular epidemiological approaches to gain a deeper understanding of the disease, the pathogens, and ultimately the epidemiology, and how we can hopefully work on that and change the epidemiology, or at least be sure that we can recognize patients at risk, diagnose them early, and treat them early. So I thought, let's start with the end. I hope that by the time that this talk is finished, or you have finished listening to it, you'll realize that to distinguish TB and NTM disease really requires diagnostic microbiology. You can't do that by eye, and you can't see it from what the patient looks like. You'll also realize that in terms of identification, MALDI-TOF is out there and works quite well to identify NTM, but as many labs are moving towards whole genome sequencing, that's what also should be done for NTM, and that will revolutionize how we think about identification, pretty simple, but also about the epidemiology of this disease. I think at the end you'll realize that using whole genome sequencing for susceptibility testing, like we can do so nicely in TB, we do it in my center for TB, really nice, that is nowhere near working for NTM. That needs a lot more work. And doing more whole genome sequencing will hopefully also learn us whether the NTM are now doing what TB has done aeons ago, and that is to become a human transmissible pathogen. So to start distinguishing TB and NTM, I thought let's take it to clinic. So this is a CT scan of one of our recent patients. And so what you see here are upper lobes of both lungs and a massive set of cavitary lesions. So is this TB or NTM? Of course, given the topic of today, you might say it has to be NTM since this is an NTM talk. But looking at the lesions, this might just as well be TB. And if I tell you that the patient is a 56-year-old truck driver of Kurdish descent who has diabetes, there are also several risk factors for TB. So the patient looks like TB, the lesions look like TB, and yet this was a Mycobacterium avium cavitary lung disease. So just judging by the radiology, especially with cavitary lesions, you're hard pressed to say, oh, this has to be either TB or NTM. Studies have looked into things like cavity wall thickness, but none of that was consistently associated with either TB or NTM. The next case, actually a patient my wife saw quite a while ago. This was a 60-year-old man of Moroccan descent who presented with an acute spinal cord injury and paralysis of both his legs. And then an MRI scan of his spine revealed this. Massive damage to his spine, or spondylodiscitis. Is this TB or NTM? Of course, we know that TB is well known to cause spondylodiscitis, Pott's disease, right? But mind you, NTM spondylodiscitis is also an established clinical entity, but it is often related to prior surgery. And very rarely do you see spondylodiscitis with NTM just out of the blue without having had prior surgery and then contamination and then infection in that area. So here NTM was less likely, and in fact, needle biopsy and PCRs rapidly diagnosed TB. Same, oh, sorry for the typo, another patient of a while ago with a cavitary lung lesion. So this was a 35-year-old lady born in Thailand who presented with a long-standing cough and then had a chest x-ray that you currently see with dense airspace opacities and this seemingly thin-walled cavity. Now, she was from a TB endemic region. She was quite young, 35 years of age, younger than what we usually see for NTM. But we do see NTM disease in these young patients. But this also turned out to be TB. But it still could have been NTM, like the very first patient, where there were also several risk factors for TB, but it turned out to be NTM disease. So moving into the lab, this is just something that I looked at through the microscope last week. A patient newly sent to our center who had auramine positive sputum. And you can nicely see the fluorescent rods, which are the mycobacteria. So can you judge by size and shape whether this is TB or NTM? No way, right? Even if you have very experienced technicians in your lab, they might say, it reminds me more of, or I think it is more of, but you can't say just on basis of what you see here, whether this is Mycobacterium tuberculosis or Kansasii or avium or abscesses. So nice, important for your clinical management, but still you need additional testing to see, is this TB or NTM? And now we get to the area where things get a little more clear cut, because this is a Lowenstein-Jensen medium slant that shows growth. And if you look at it, you see it's a bit glossy and nice dome-shaped colonies, maybe slightly pigmented. So here, I think everyone who's spent some time in the mycobacteriology lab, if you'd ask them, is this TB or NTM? Would say, well, it's extremely unlikely that this is TB. This is way more likely to be a non-tuberculous mycobacterium growing on the LJ slant. So a lot of these show that with a combination of clinical factors, radiology, you can sometimes guesstimate where things are going, but you can never be sure until you've seen the evidence from the microbiology lab. And interestingly, another group where this distinction between TB and NTM is sometimes a little difficult is in children with long-standing lymphadenitis, so subacute lymphadenitis. And Aina Martinez recently published a really nice paper with a large group of collaborators from many different countries, looking at cohorts of children with NTM lymphadenitis, mostly Mycobacterium avium complex, and TB lymphadenitis in kids. And then they analyze, so what are the differences? Can you see or guesstimate whether something is TB or NTM? And they found some interesting things. So the kids with NTM lymphadenitis tend to be younger than kids with TB lymphadenitis. So the NTM patients were usually below the age of 5. Also, NTM lymphadenitis was more common in children who are born in low TB endemic settings and, accordingly, are not BCG vaccinated. Also, the NTM kids were typically not suffering from other diseases. And again, diagnostics, TST, so skin test positivity, was less frequent among kids with NTM lymphadenitis at 60%, versus those with TB lymphadenitis in whom 98% had positive TST. The difference was even bigger, and I think we can all understand that, for the interferon gamma release assays, or IGRAs. Out of the cohort with kids with NTM lymphadenitis, only one child had a positive IGRA, whereas among the cohort with TB lymphadenitis, 91% of those children had a positive IGRA. So subacute lymphadenitis and a positive IGRA will really set you on a track towards, this is quite likely TB, pediatric lymphadenitis. So that was a really cool finding. And the other aspect that was quite interesting from an etiology point of view was that TB lymphadenitis affected mostly cervical lymph nodes, whereas for the NTM, it was a little bit cervical, but a lot of it, like the picture you see on the slide, was submandibular. And that, of course, may relate to how the bacteria are contracted. So, to make distinctions between TB and NTM, you need diagnostic microbiology. And you can't judge it from where the patient's from, or how old the patient is, how the patient looks. Okay, so now that we've said that the lab is so important, we should touch upon, okay, so what's new in the lab? I trust that the pictures at the bottom to many of you will be somehow familiar. Tubes with sputum specimens in this case, go into the lab, are processed for microscopy and both broth and solid culture. Well, a lot of that stuff we've been doing for ages and ages. So what's new? Well, it's the things that come after culture that are new. So to identify NTM to species level in itself, well, it's not entirely new, but I think that the understanding that species identification is essential to make sense out of the result in clinic is something that is now widely established. So I really hope that no lab will ever again say, oh, we cultured an NTM to their clinicians. You identify it to species level because only then you can make sense out of it. So in the last few years, MALDI-TOF has emerged as a new and widely available method to identify NTM to species level, and protocols to do so have been steadily improving over the years. And there's a really nice paper by Al-Qaeda, again, from a large group of collaborators who published a nice how to do it paper in the Clinical Microbiology and Infection journal that I recommend anyone interested in doing MALDI-TOF for NTM to read, because it has great hints on how to do it. And when I explain it to pulmonologists in training, I always explain it like, look, if you put a tomato on a table and just smash it and look at, okay, so what does the mess that I just made look at? You can kind of guesstimate what type of tomato it was. And that's how MALDI-TOF works, be it in slightly more detail. The other thing that has changed in last years is the identification of NTM by molecular means. So more and more labs get access to DNA sequencers. So while identification for long relied on probe-based assays like line probe assays, more and more labs now have access to sequencing facilities and can either sequence one or perhaps two target genes or sequence the full genome to come to an identification of isolated NTM. In our center, we are now routinely using whole genome sequencing, and I can tell you that has really changed a lot for us. So a lot of things that we thought about, in very simple terms previously now show to be quite difficult, like, in things that we previously called the Mycobacterium fortuitum complex, we now identify to species level, and we see there's a lot of variety. So from there, we can learn and see whether some species are, for example, linked to particular diseases or disease manifestations. The same goes for MAC. And we also learn that some published species are so close genetically that it's almost impossible, even by whole genome sequencing, to distinguish those. And maybe taxonomists should think or rethink whether such species are not merely subspecies. So also for taxonomy, I think this kind of data is really helpful to rethink the current taxonomy of the non-tuberculous mycobacteria. The other great thing about whole genome sequencing is that if you find something that is potentially new, like the isolate that we sequenced last week, which I put the graph in here from our phylogenetic pipeline, which was sort of at the edge of the avium complex and scrophulatium group. So you can immediately see, okay, what's it like? And sometimes that's helpful to think about if it's, amidst a group of well-known opportunistic pathogens, that potentially new species might also be a potential pathogen. Whereas if it's among, a group of NTM that we only know from environmental studies and that we don't ever encounter in clinical practice, then that might also reassure you that this one also is most likely not that pathogenic. So I think the era of whole genome sequencing is now here. More and more labs have access to it. We know how important it can be for TB. And I really hope that more and more people will also use it for NTM to also allow us to study the genomes of NTM, because we don't really know that much about them. And also be able to compare what is Mycobacterium avium in the Netherlands, compared to what we call Mycobacterium avium in Germany, in India, in the United States, in Argentina. How closely related are they? And what are differences? So there's a lot that we can learn, and I really hope that that's going to really get going in the next few years. Well, as I said, the other thing that we know works well with whole genome sequencing for TB is to predict drug susceptibility. Well, very few people do that for NTM, so we're still learning. That's one major development, I think, in DST of NTM. The other major development in drug susceptibility testing for NTM is that UCAST has finally picked up on Mycobacteria. So there now is a reference method for Mycobacterium tuberculosis, and a reference method for the NTM is currently in development. And I think that is really, really the right way to approach this. Start from the basics. Set up a reference method, start working on ECHOVs, start studying resistance mechanisms, and then take the next step, which is also currently coming up, and that is to include PKPD science. So to use models like the hollow fiber model or animal models, although I personally think animal models for TB are not that good, and then see how these models can help us to set PKPD informed clinical breakpoints and really show what is drug susceptibility in NTM, because that is still a very tough subject today. So, as I already alluded to, the use of whole genome sequencing to predict drug susceptibility is in its infancy in NTM, very different from TB. The problem is that for many of the drugs that we find important in clinical practice, we might understand the primary mechanism of resistance for most of them. Secondary mechanisms are by and large unknown. And I think if we follow the isoniazid example in TB, where we know that several mutations in several genes and systems all lead to increased MICs or resistance to isoniazid, that is something that for the important drugs in NTM treatment, we simply do not know. And it will take a lot of whole genome sequencing, I think, to learn about these secondary mechanisms, but also other omics studies. So we're currently involved in a lot of transcriptomics studies to look at what networks or what genes are activated or repressed upon drug exposure in NTM. And hopefully that will lead to understanding a lot of secondary mechanisms that we can then screen for mutations. So knowing what we currently know, studies have looked at predicting amikacin resistance, for example, using the 16S mutations that we also know from TB. But doing so, the sensitivity of detecting resistance by these means is only as low as like 68% in one very well-performed study from South Korea. And the senior author of that paper, Dr. Hyun-Woo Yun, is on the picture there. And two other studies have looked at predicting macrolide resistance in abscesses using whole genome sequencing. And that was work by Samuel Lipworth, who is presented by the cartoon at the bottom. And they showed that the sensitivity of whole genome sequencing to predict resistance was quite good at 95%, but the specificity was very low. So there was over-calling of resistance. In the Mycobacterium avium complex, calling macrolide resistance from genomics was easier, and the sensitivity of using sequencing approaches of the 23S gene had sensitivities in the upper 90%, so 96% in one study. And that is something that we also recognize from our practice. So there are some things you can do in predicting drug resistance for NTM from whole genome sequencing, but we have a long way to go to make that into a diagnostic test that then is so good that we do not need the phenotypic tests anymore. We're not near there yet. So MALDI-TOF is a reasonable identification method, but whole genome sequencing, I think, will revolutionize identification and also epidemiology of NTM. And to use it for DST requires a lot more work. We're not nearly there yet. As I said, sequencing approaches are important for the epidemiology, and that's what I think the last few minutes should be spent on, because I think that is where the big changes in our thinking about NTM and how they cause disease is really going to be affected over the next few years. So I'd like to end with the third part on evolution and mutation in NTM. And well, sadly, it is still into the great unknown, but I think I'll touch upon a lot of things that will, over the next few years, be either confirmed or deepened, and it will change our understanding of these pathogens and the disease that they cause. So some very initial work that we've done within NTMnet with my friend Dirk Wagner from Germany and my friends from South Korea and Japan and the colleagues at National Jewish was very simple. We collected a series of Mycobacterium avium isolates from our labs, sent them all to our friends in Japan who did VNTR typing, and guess what? There were very different genotypes, and strains coming from the same part of the world tended to combine into the same genotypes. So strains from Japan and South Korea were more closely related to each other than they were to strains of geographically distant sites like, the United States and Europe, and the other way around. So this suggests, and I think that makes sense, that there are distinct Mycobacterium avium ecotypes worldwide. The question is, do these different types also have different virulence or lead to different disease manifestations? That, of course, is also a matter of the host, but that is something that you can only study once you realize that there are different types. And how different the types are in your own local area is something that I think still has not been investigated properly. So if you zoom in a little more, so we go to individual patients, genotyping efforts, again, by and large by VNTR typing, of which the discriminatory power in NTM is suboptimal. We've already learned that for some, not all, patients, an environmental source, usually in the home environment, of their infection could be established. So studies in the United States and Japan have linked patient isolates to NTM isolated, mostly avium again, isolated from their bathtub or their household plumbing. And also interesting, especially in these COVID times, now more and more people have reinvented gardening as a hobby, including myself. NTM cases have been linked to NTM isolated from potting soil that these patients had used during gardening. And in Australia, there's an interesting and ongoing story of Mycobacterium abscessus isolates in one area, so clinical isolates that were genetically highly similar to isolates from the local water reservoir, so the public water reservoir, which was first thought to be the source of infection. But later on, that was questioned because maybe the water reservoir was contaminated by a human carrier of the abscessus. So whether it's water to patient or vice versa still remains to be established. But molecular typing has shown that for some patients, you can find the likely source of their infection. And then, as I said, the other interesting area where you can use typing is in the patients who you treated successfully for their NTM disease and then get a recurrence. And then you can establish whether it's the same isolate again, suggesting but not proving that it's a relapse, or whether it's a different strain altogether, suggesting a reinfection. Of course, if it comes from the home environment, reinfection with the same strain is also possible. But the interesting thing that our colleagues in the United States, in Tyler, Texas, found a few times, first with classic PFGE typing and later with VNTR, is that patients with the nodular bronchiectatic disease who have a high recurrence rate mostly had real clear reinfections with different strains. In the patients who had cavitary disease, a lot of the recurrences, sadly, were relapses with the same strain. So why is that important? Well, if you find that it's a relapse with the same strain, you've not been able to truly sterilize the lungs of these patients, and that means that your treatment is failing. So that also means that you might have to go into a more intense treatment regimen for this relapse of the disease. Whereas in reinfection, essentially you start all over again, so you can start with first-line treatments probably. And then our colleagues from Japan took it one really nice step back, and they thought, well, is it maybe possible to predict the disease course from the mycobacterial genotype? And of course, in opportunistic infections, the host, of course, is incredibly important. Nonetheless, they did find some associations between Mycobacterium avium genotype and the course of disease over time. So fascinating. I think in the era of whole genome sequencing, we really need to repeat this work. But it is just fascinating. So that maybe there is a mycobacterial component to how the disease presents and behaves. So the very last story, and I hope I can still use the last minutes of attention that you have left, is, of course, I must say, about Mycobacterium abscessus and the human transmission story. You may have heard from it, you may have read it. Fantastic publications by Andres Floto and Josie Bryant and their group, first in the Lancet, about a single center outbreak of Mycobacterium abscessus among cystic fibrosis patients. A follow-up paper in Science in 2016 showing that in countries around the world, there are very similar Mycobacterium abscessus strains present. And in fact, showing that the majority of patients in all countries have isolates that are identical or near identical to strains of other patients, both in their own region as well as abroad, suggesting, again, human transmission, maybe indirect transmission, but at least transmission, and 3 very dominant clones worldwide. So the fact that they are potentially transmissible from one CF patient to another, of course, is worrying and has totally changed how we see CF patients in our center in terms of infection prevention and control measures. But the troubling thing is that they also saw that the strains involved in transmission chains were more virulent, were more drug resistant, were better able to survive in macrophages, and as I said, more virulent in mice. Very troubling. And then a few weeks ago, a paper came out in Science that put the next step. And again, the authors, it's the same group, show by genomic analyses how maybe Mycobacterium abscessus, maybe Mycobacterium avium, is perhaps on its way, like TB, to become a human transmissible pathogen. And they show that mutations in transcriptional regulators have probably shaped the dominant clones that we now see spreading. And that if you see within the host, so within that CF patient, over time, the changes in the mycobacterium lead probably to better being able to survive in macrophages. And these can then be transmitted via the environment to the next patient. So what we're, well, waiting for, I hope it never happens, is the next step, that these bacteria will change so they will retain their virulence, but also become directly human transmissible. So I recommend Josie Bryant's latest Science paper. So it's a brilliant read, but very worrying. So, as I said at the start, I hope you now realize that you can't distinguish TB and NTM disease from just looking at the patient. You need diagnostic microbiology. The labs should really look at identifying NTM properly. MALDI-TOF is a nice first-line method. I hope sequencing approaches will take over, but not to call resistance yet. And then, as I said at the end, we should really work on typing NTM to better understand the epidemiology of these diseases, and to see whether the Cambridge group is right, that some NTM are, like TB, moving towards becoming human transmissible pathogens. Well, to close down, I'd like to say if you want to learn more about NTM, there's a fantastic NTM slide repository at the NTMnet website, and the Embark website that you can have a look at covers everything NTM. And these are the wonderful people that I work with at the Radboud University Medical Center. So thank you very much for your attention, and I hope you have cool questions.
Thank you very much for this very interesting talk, opening our eyes and opening our minds in thinking of NTMs also in the future, in this evolution. There's only one question I can see at this moment in the chat, and this is from Abigail Perez from the Philippines, and wondering whether patients suffering from NTM infections are given the same treatment as patients suffering from pulmonary TB. So it's about treatment.
Well, that's a brilliant question. Of course, the answer is no. So antibiotic treatment of NTM disease should be very different from TB treatment, and it very much depends on which NTM species is causing the disease. But as I said, since it's so hard to distinguish TB and NTM disease when the patient first presents, what happens a lot, of course, to break potential chains of transmission is that you start TB treatment while you are awaiting the microbiology results. And, the patient put in isolation if you think of pulmonary TB, and you may start TB treatment. And then if you find out, hey, it's not TB, it's NTM disease, you need the species identification, and preferably also drug susceptibility testing to then choose what is the antibiotic treatment regimen that my patient needs.
Okay, thank you. Maybe connected to that, so you recommend either MALDI-TOF or whole genome sequencing for the identification of the species, which is not available everywhere. But I was wondering the turnaround time for your whole genome sequencing. Is that routine practice already, and what's the turnaround time?
Yeah, so in our center, whole genome sequencing is now routinely applied. So we perform it once every week. We hope that if the per strain cost goes down more, because you have to use large chips, which you can sequence 100 isolates at the same time if you want, but then we don't have 100 new positive cultures in a week. So yeah, we hope if the costs go down, we can do it twice weekly. Of course, there are a lot of nice in-betweens. So there are a lot of molecular probe assays that you can use to identify the NTM, Lyme probe assays, for example, that I think provide really quite clear results that are definitely easily used in clinic. So, of course, if you have access to sequencing, it's more exact. But MALDI-TOF or Lyme probe assays, I think, are really good ways to identify NTM and give you results that you can easily use in clinic.
Okay, there are some questions from Daniela. She asks to comment on the rise of NTM in cystic fibrosis patients and how accurate are the reported data? Can we really rely on the correctness of these data or?
Yeah, of course, NTM pulmonary disease or any NTM disease it's not a reportable condition with a few minor exceptions in one province of Australia and one state in the US. So accurate epidemiological data are lacking. However, there are consistent reports, including in cystic fibrosis patients, that number of people with NTM infections is on the rise, but that has several reasons. One of them is that CF patients nowadays tend to get a lot older than they used to do. And particularly the MAC infections, or Mycobacterium avium complex, we tend to see at later ages. So there's new groups at risk. And the same goes in the non-CF population. I mean, a lot more people nowadays use immunosuppressive medication, which renders them at risk of opportunistic infections, including NTM. And also, sadly, because of smoking habits, the prevalence of chronic pulmonary disease like COPD is on the rise. The prevalence of bronchiectasis is on the rise. So more and more people are at risk of contracting NTM disease. So are the data solid? Not that solid. This is not TB. It's not a reportable disease. Do I think NTM disease rates are increasing? Absolutely. Yeah, they are.
Okay, clear answer. And a question from Chiara from Argentina. Which is the algorithm to use for MALDI-TOF? I think the question is when do you, it's on positive cultures you use it, or I don't truly get the question, but.
Yeah, well, of course you use it on positive cultures. And then there are various extraction methods. So for example, the Bruker company that produces one of the machines, they have an extraction protocol, which I think is called MycoX, works really well. There are also really nice sonication methods described by the Al-Qaeda et al. Paper in the Clinical Microbiology and Infection journal, I think 2 years ago. Those are really easy to use and really effective strategies to do the extractions before going into the MALDI-TOF machine. And then you need a good database to make sense out of it. Both the big companies producing MALDI-TOF machines nowadays have quite good databases. I think the Bruker database used to be ahead of the other one in terms of how many species it had and how many isolates, but I think both are now sort of at even levels. Okay,
thank you. Tim Bull is questioning whether you feel that latent non-tuberculous Mycobacteria infection, that states also exist. Do you have latent NTM infection in the same way like you have latent TB infection? And what's the prevalence of that? Is there any data known on this?
That's a really, really, really difficult question. And I applaud Tim for asking that because we simply don't know. What we do know, of course, is that if you use the NTM skin tests, you find skin test reactivity, suggesting at least that people have seen it. But whether there is something like a latent infection, I doubt that. But the problem, like in TB, is how do you show it? How do you show that there is a real latent infection? I mean, even in TB, I think that's a hot topic of discussion today. In NTM, I would say probably not, but in all honesty, we don't know.
No, true, difficult to study. And this may also evolve in future when they become like true pathogens, of course. Yeah. Another question from Daniela is, probably following on this cystic fibrosis cases, do you think NTM should become notifiable diseases?
As an NTM enthusiast, yes, but we have to think about what will the impact be of making it a notifiable disease. I think it should be done, at least as a pilot experiment, to see the real prevalence of isolating NTM from patients and how many of those isolates actually represent true disease. So in that sense, I think, making it a notifiable disease, or at least for a short amount of time, I think is incredibly useful to really understand the epidemiology. Of course, unlike in TB, we're probably not going to set up a control program, because how are you going to control NTM? It's not human to human transmission, so we can't prevent people from being exposed to NTM. So there are very little actions that you can take after you realize from your notification rates that something is going on. If you say, oh, the avium rates are going up, what can you do? Disinfect your local environment. So I think it's important to understand the epidemiology, but I think there are very few practical consequences. Nonetheless, I feel it should be done.
Yeah, I agree. Has NTMnet set up a kind of possibility to notify or to register NTMs? Is that something you are working on or emphasizing?
Well, not at this stage. In the Netherlands, though, we are working on this together with our friends at the TB reference lab. We are trying to collaborate on NTM and also make a case to at least have a pilot period where we notify NTM isolation. But that, still we are discussing that with the political side of things.
So this is something that could be considered at this moment at country level as a research project, indeed. Yeah. Okay. I think we are almost getting to the end. So regarding the relapse and reinfection, you have shown this analysis of the Mycobacterium avium example with the classical RFLP or AFLP. I don't know what you did there, but is there any practical typing technology available at this moment? It's different for each species, and I think whole genome sequencing cutoffs have not been defined yet, or what's your thought on that?
Well, yeah, both I think are absolutely true. The problem with NTM, very different from TB and other infections, is of course that they are NTM. They are a grouping of 200 different species. So if you want to look at typing, you either have to use a generic approach like whole genome sequencing, which of course is incredibly expensive. It's hard to get access to, but well, I mean, slowly it is. Access to that technology is expanding. The other option is to indeed focus on one particular species. And that is when you can also start using other techniques. I mean, in the past, for Mycobacterium avium, RFLP has been used, using the IS 1245 or 1311 insertion sequence. AFLP has been used as a generic method. So there are various methods that you can use, but also depending on the question, you have to look at what kind of discriminatory power do you need, and then select the method on basis of that. But I hope TB reference labs across the globe will be moving towards whole genome sequencing, and then will allow NTM enthusiasts like myself to collaborate and also work on NTM in there. I mean, this technology is brilliant, but it's so expensive. We need to use it as much as we can, because that is the only way to lower the cost, is to run more samples per run. So work on NTM if you have the technique for TB.
Yeah, I think that's a perfect note to close this session. And we really want to thank you, Jakko, for this very interesting webinar, and also thank the sponsor again. And we also look forward to meet you all in the next webinar. And of course, also in our next online conference that will take place, for which you can still register. So for the webinars, no need to register, but don't forget to register for the online conference. I think I have nothing else to add to this. So big thanks to you, Jakko.
Thank you for the invitation, and everyone stay safe in these odd times. So thanks a lot.
Bye.